All right, our first video on protein synthesis. So our essential questions: how is DNA's code expressed? Number two, how can a change in DNA's code affect living things? Your skills, something you have to be able to do by the end of this video, is if I give you a nucleotide sequence of DNA, you have to be able to determine the sequence of base pairs that would be on the opposite strand of DNA. So DNA is complementary. If you know what one side of the strand reads, you should be able to tell what the other side reads. Now, protein synthesis, or the making of proteins, follows this idea of the central dogma. So we know DNA is a molecule that stores information, but the information that it's actually storing is how to make proteins. But remember, DNA is kind of like an instruction manual that you want to keep safe. So we keep it stored away inside the nucleus, meaning that DNA is not really going to get out and do its own dirty work for actually making any of the things to sort of run you. That's what it has RNA for. What we're going to focus on in this sort of video is looking at DNA. So if we were to take it back to genetics and talk about it, we would be looking at genotype, so what's in the DNA. Whereas what we're going to end up with proteins, that's phenotype. That's what you actually see. So a little history on DNA. DNA stands for deoxyribonucleic acid. It was discovered in 1897 by Friedrich Michner. However, the structure of it was really a mystery for a very long time, and it wasn't until 1953 in the edition of Nature that a paper was published with these gentlemen right here, Francis Crick, James Watson, and Maurice Wilkins, publishing a model, the first successful model of the structure of DNA, and here it is right here. Now, the sort of darker side to the story is that they would not have been able to come up with this discovery of the structure of DNA without the help of this woman, Rosalind Franklin, who was really an unwilling participant in this discovery. Her lab, she shared a lab with Maurice Wilkins, who thought she was coming to King's College, where they all worked in England, as his assistant. She was coming there to complete her own work, so the two had a very contentious and negative relationship. Wilkins actually stole some of Franklin's work. She had taken an X-ray of DNA, and he gave that picture to, known as Photo 53, to James Watson and Francis Crick, and it is with that that they were able to figure out the structure. They won the Nobel Prize, and she was never recognized formally because by the time that they had won, she had already passed away. So here's your double helix structure. It has two sides, a sugar phosphate backbone that runs along the outside, and then your base pairs or nucleotides that are going to create the rungs sort of on the inside here. Structure is often referred to as a twisted ladder or as a spiral staircase. So we have to look at a little bit at how DNA is organized. The largest scale, so we're going to go from largest to smallest, is your genome. Your genome contains all of the genetic material of any organism. So the human genome, which was sequenced fairly recently in the last couple decades, just looked at what's all of this, what's in every single one of our 46 chromosomes. Now, when we look at the genome, we're talking about coding regions. So these are sections of DNA that actually have information that's used by your body to create proteins. And then there's also junk DNA, so sections of DNA that don't really code for anything. They don't have directions. Some of it can be leftover DNA. Some of it can be viruses that had sort of invaded and sort of stuck themselves in your genome a very long time ago. There's a lot of kind of stuff in there that's not used. So your genome is made up of chromosomes. Chromosomes, here's a replicated one with its two sister chromatids, are the structures of tightly wound and organized DNA. Humans have a total of 46 chromosomes, which comes in 23 pairs, one from mom, one from dad. In your chromosomes, there are regions of DNA sequences known as genes. These are actually pieces of DNA that contain the directions for how to make a protein. One gene equals one protein. So every gene has the directions for making a single protein. The number of genes are going to depend, not always, but typically on the size of the chromosome. But chromosomes contain hundreds of thousands, if not thousands, to thousands of genes. So there's lots of genes on every chromosome. But remember, not all of the DNA in a chromosome is actually going to code for proteins. So your genes are made up of sequences of nucleotides. So nucleotides, you may remember from when we did organic molecules, they are the monomer of your nucleic acids, which is what DNA, nucleic acid, and RNA, nucleic acid, are. So you remember there are three parts that make up a nucleotide. Both of these right here, here's DNA. I can tell it's DNA because I see the deoxyribose sugar base, and I can tell this is RNA because I see the ribose sugar. Both of them have phosphate groups, both of them have 5 carbon sugars, and then both of them have nitrogenous bases. Now, the phosphate and the sugar stay the same regardless. However, these nitrogenous bases are going to change, and this is what actually is going to change in the structure of your DNA, and it's going to give you this huge variety of DNA coding that you actually get. So when we talk about the sequence of DNA, we're actually just talking about the order of the nitrogenous bases, so these last pieces right here on your nucleotides. And here are your nucleotides. So for DNA, there's 4 nucleotides. There is guanine, cytosine, adenine, and thymine. Now, these always pair up in a very predictable way. So if you remember your skill from the beginning, I said that if I gave you one side of DNA, you had to tell me what's on the other side. So you see right here, guanine and cytosine, they always pair up. So if I tell you one side of cytosine, well, you know cytosine only bonds with guanine, so the other side has to have guanine, and it works vice versa. Same thing with adenine and thymine. Now, remember, we're just looking at the nitrogenous bases, so each of these is going to have a 5 carbon sugar and a phosphate group attached to it on either side. This is really what makes up the rung or the inner part of our double helix. Now, RNA is a little different because it has, instead of having thymine, adenine is going to pair up with uracil instead. So whereas on a strand of DNA, if you see thymine on one side, you know it's adenine, or see adenine on one side, say it's thymine. If we're looking at RNA, if you see adenine and you write thymine, that's not right. No thymine in RNA. You'd have to know that it's uracil. So base pairs are held together by hydrogen bonds, which makes them relatively easy to break, because if you remember, your hydrogen bonds are fairly weak. And we have 2 types, so guanine over here and adenine with 2 rings are known as purines, and your single-ringed nitrogenous bases, thymine, cytosine, and uracil are known as pyrimidines. Purines always bond with pyrimidines. So an overview of the difference between DNA and RNA. This hopefully should be a review at this point. Remember, DNA is double-stranded, RNA is single-stranded. You know, you have different 5 carbon sugars that make up your nucleotides in the 2. So DNA takes deoxyribose and RNA takes ribose, which is why we get this difference in the first letter of our acronym. And then last but not least, this is most important for being able to order and sequence these. Thymine is only in DNA, and uracil is only in RNA. So when you're looking for who adenine is going to pair up with, if it's DNA, it's thymine. If it's RNA, it's uracil. So the best way that I have to remember the levels of organization of DNA is to compare the different levels to a book. So if you think of your genome as being a book, each of the chapters of the book would be like the chromosome. So the human genome is made up of 23 chapters, 23 different chromosomes. Now, within each chapter, we know the chapters are made up of paragraphs, just like your chromosomes are made up of genes. Now, whereas books are written in different variations of 26 letters, the genes are made up in variations of only 4 letters, A, T, G, and C, because these are the 4 base pairs in DNA. You can make hugely variable combinations with just 4 letters, though, which is why we're able to have so many different proteins. So a little practice. Identify the following strands as DNA or RNA. So strand number 1, DNA or RNA. So hopefully you were able to get that this is DNA, and I can tell that because what do I see right here? I see thymine. Big hint. Strand number 2, DNA or RNA. Good, so hopefully I just gave you a big hint there when I said number 1 has thymine in it, because if you look down here, do you see any thymine? No. But I see uracil, and I know uracil is only found in RNA. Last check. All right, write the base pair sequence of DNA that would complement the following strand. So if I give you that 1/2 of your double helix has a thymine, guanine, cytosine, cytosine, adenine, thymine, thymine, guanine, cytosine, what's the other half have? Good, so hopefully you were able to get this strand right here as being the complement. So adenine and thymine always pair up, cytosine and guanine. So 1/2, you can always tell what's on the other side. So make sure you filled in all your guided notes. Send me an email if you have any questions. And remember, at the end of this, you should be able to, if I give you a sequence of DNA, tell me what the other half of that DNA strand would have just based on the base pair sequence.